Hello and welcome back. My name is Anders Larsen. If you didn't see the sampling intro, you may do it at this point in time. Otherwise, you're welcome to just stick around. Battling the challenges I mentioned in the intro is actually quite easily overcome because there is only one rule we have to obey in sampling. That is, all particles in a truck, a ship, a field, a tank, a pipe must have the same non-zero probability of becoming part of the analytical result. That is very easy to say. It is quite a challenge in real life, but let's dive a little deeper into the subject. When we deal with sampling, the first thing we look at is the dimension of the challenge. We have three dimensions that we look into. We have the three-dimensional challenge. That would be a ship, a truck, a tank. We have the two-dimensional challenge. Let's keep it here. The 2D would be a field. And then we have also a one-dimensional, which is a pipe. Once we understand that these dimensions are present, we can now revisit the first rule. Are all particles equally accessible to us when we take a sample? From the tank, either we would take it from the top, hence the bottom part of the particles would have a zero probability of becoming part of the cup. If we have some kind of an outlet, only the product close to the outlet will get part of the sample. Likewise, if we have a field and there's a small road, we park the truck somewhere, and generally speaking, the products close to the car will have a slightly higher chance of becoming part of the sample than what we see if we really were to do it random. There is a theoretical 0D, which is a very beautiful construction, but in real life, the way we want to go is to see, can we transform these dimensions into 1D? This tank was filled at some point, and it will be emptied at some point. This might be a harvester, and the harvester will put stuff in a truck. So quite often, things in a 3D or 2D will pass a pipe at some point, and every little particle in this 3D or 2D will have to pass a very narrow, one-dimensional, pipe-ish kind of construction. And in this, we can take out random, meaning every particle have the same probability of being caught in the sample. So that's the first thing. There's a vocabulary here. So we talked about the sampling dimensionality. And when we take out a sample, we do not talk about a sample until it's ready-made. I'll come back to that. Taking out something we call an increment. So these are some of the words associated with the initial thinking of physical sampling. The next thing we want to look into is heterogeneity. We have basically two types of heterogeneity that we want to battle. If we look at a cross-section of a pipe, we may have the fact that we have equally many blue and red particles, but they may not be distributed equally. Larger, smaller particles, something that flows, something that doesn't. So no matter where we take one single sample, this compositional heterogeneity is a challenge for taking a single sample. If we turn this pipe around, we will have that there might be at some point a lower concentration of blue, but there might be a higher concentration of red. This is what we call the distributed heterogeneity. So if we want to know straight away how much we have, and we take out a sample, and we think this is a sample, and we do that here, and here, and here, we will conclude that the amount of blue is near 100% and is completely stable. So this is of course not true. We will have simply to find ways to battle these two heterogeneity challenges. The simple answer in fighting these two things is relatively simple. For compositional heterogeneity, we simply move away from a single sample scheme and we take out multiple sampling. That means composite sample. So we will take out a sample and a sample and a sample and a sample and a sample, and we may take 5 or 10 small increments, and the chance that we get all around the plate is clearly that we will get a much better average. Likewise, over time, we will have to take out samples on a belt or a whatever it is. And in this way, we will not judge this process by this sample or this sample or this sample, but simply we will create a population of observations and then only after that judge. So no more taking one simple sample, because the rule of thumb is that grab sampling becomes crap sampling. Once we have taken out a composite sample, that sample is quite often too big. If we think of a ship, we may take out 50 kilos or 100 kilos in order to represent a ship carrying hundreds of thousands of tons of product. It seems like really annoying to have 100 kilos. So we better reduce this, otherwise we can't get to the lab. But here, mass reduction, representative mass reduction, is another topic that one needs to educate himself in, and there are loads of articles. So mass reduction to coming from composite sample mass to the analytical mass, keeping in mind that most reference methods will look at very few grams, like a protein determination. Think about 1 gram for a kildal. We come from a ship of hundreds of thousands of tons. We want to pick that 1 gram that tells us this ship is. That's not trivial. And very careful representative sampling, composite sampling, mixing, and other unit operations and representative mass reduction is essential. In the next graph here, we can see that sampling errors have been organized. This is a graph taken from the standard DS3077 that I mentioned earlier on. We see that we no longer talk about analytical error. We talk about global estimation error. The global estimation error is a sum of total sampling error and total analytical error. The total sampling error is the hidden gem in understanding why results are not always the way we want them to be. Total sampling error is typically far greater than the total analytical error, being the repeatability of a reference method or what have we. These errors have been organized in steps showing that primary sampling is the step where we take it from the ship. If we then subsample that and split it into 2, that's another sampling step. And finally, the last step could be sampling in the lab, where we take out a small portion. The important thing is that we can repeat all the same mistakes in all steps, and it will add up to the total sampling error, keeping in mind that the largest error is typically done in the first step. Some of these errors can be avoided, some not. In the theory, it's called correct sampling errors and incorrect sampling errors. I prefer the avoidable and non-avoidable. The unavoidable is simply that we have fundamental sampling errors. You cannot take the same sample once you have done it here, and you have grouping and segregation as a natural part of life. These just have to be well understood and characterized. But there are a whole bunch of errors that can be eliminated, and these are well described in the standard. I will not get into them here, but they are dealing with tools, how we pack the sample, how we deal with the sample, how we delimit, and that is very, very important to do that right. Okay, welcome back to a summary of the introduction to the practical aspects of sampling, which summarizes down to 3 principles and 4 practical procedures. I always recommend the first principle: characterize the heterogeneity. Know the beast in front of you. If you have something fairly with a low degree of heterogeneity, of course you can take fewer increments, whereas if you have something very, very heterogene, you would want to take more. But know the beast in front of you. Likewise, for online stuff moving, do variographic studies. Know how much does it actually vary. I refer to the next video in part 2 where we'll dig further into that. And always remember to reduce the dimensionality from 3 dimensions into 1 dimension. Much easier to pick it out when there's an equal probability that you can catch it all. Then unit operations, mixing, blending, reduce particle size, always do composite sampling, and do representative mass reduction. Then you have done a lot of the things needed to do a very, very good sampling and improvement over what we commonly tend to do. In the next video, I will talk about sampling in time. Hope to see you there. Thank you for now.